We've now done a lot of whole genome sequencing in leukemia, and we found that most of these patients, kind of like that circle that I showed you earlier, have very few mutations. And there's already sort of a paradigm for disease classification and making treatment decisions on a new AML patient, right, that uses the genome. You basically use the genome by looking at chromosomes, like I just said. So when a new patient comes into the clinic, has signs of leukemia, a bone marrow biopsy is done. You send some of that material to the pathologist who looks at it under the microscope, right? He's basically trying to look at the cell type to say, I think it's this type of AML because I see immature promyelocytes. So that information is useful to the treating physician. The cytogeneticist looks at the chromosomes and says, I see a translocation, part of chromosome 15 is broken off and gone to chromosome 17. That's useful information to the physician. He knows what kind of drugs he can give the patient to give them a good chance. But in the case where the cytogeneticist doesn't see any chromosome translocations or breakage or additional chromosomes, he really can't provide all that much information back to the treating physician. And so that's what we want to know. This just shows you all of the kinds of things that the cytogeneticist might tell you. This is sort of the vocabulary of the molecular biologist, but for example, T1517 indicates a translocation where part of chromosome 15 and chromosome 17 have swapped. That's actually a good indicator. If you see that in a patient's cytogenetics, you know what drug to give them. 25 years ago, that patient would die. Now we've got a drug. 95% of patients with that particular chromosomal aberration can be cured. And we'll talk a little bit more about that in a minute. Well, what we really wanted to do was to dig deeper. Because if you do that sort of cytogenetic analysis, you look at chromosomes, right? You can find some things like the one I just told you, 1517 translocation puts patients into this bin. And this is a survival curve, a Kaplan-Meier curve to show you that that these patients with a favorable cytogenetic profile, basically 50% will survive for 14 years, which is pretty good for acute leukemia. There are some patients that you will find things in their cytogenetics analysis, and they have a very poor risk. You can see here that a great bulk of them won't survive 2 years post-diagnosis. But most patients, 61%, sort of fall into this intermediate group. And this includes both good outcome cases and bad outcome cases, but they're all cytogenetically normal. We don't see anything in the cytogenetic scan, and so we really can't say anything about them. And so most of these patients are treated very aggressively, which if they're older or in frail health, it's pretty hard on them. The cure can almost kill them more quickly than the disease. It's pretty aggressive chemotherapy, and we'll talk about that in a minute. So you'd love to be able to know for most of these patients, are they good outcome or bad outcome before we start treatment? If you know, they had a sort of a good outcome situation, we could be a little less aggressive in the treatment. If we knew that they were, you know, they had a really bad genetic type of AML, we might go right to bone marrow transplantation. So that's what we'd like to know. So tell you a little bit about the treatment that these patients receive. So when a patient is diagnosed with AML, he is immediately admitted to the hospital and given 2 very aggressive chemotherapy drugs to try to induce clinical remission. Essentially, these drugs are killing off all growing cells in the body. So not only those stem cells that would differentiate into cancer cells now, but you know, cells that grow hair, lining of the mouth and stomach, and so on. So it's a pretty nasty treatment. It's also expensive. For each patient with the hospitalization, it's somewhere between $500,000 and $800,000. In some cases, like the patients that have a type called M3, where they have that translocation I told you earlier about, we know what's going on there. There are 2 genes that are fused, and we simply give them this drug called Atra, which costs about $50,000. And these patients will essentially be cured. They'll be cancer-free within a year. So you'd love to be, and we have a way of finding that. We simply look for that translocation. But for the patients that have very high-risk cytogenetics or that essentially don't progress with this initial kind of treatment, Quite often you'll do bone marrow transplant to try to save their lives. This is a dangerous procedure. They're going to be immunocompromised for some amount of time, and it's expensive. It can add up to another $250,000 to $500,000 of health care costs. So you'd really like to be able to dig down and to better understand, especially for this middle group of patients, what's their risk. And this is why we started sequencing genomes. So this actually shows you leukemic cells from the first patient whose genome we sequenced back in 2007, 2008. This was a woman in her mid-50s. At the time she was diagnosed with AML. She had a family history of both AML and leukemia. A really aggressive case. All of the cells in her bone marrow were essentially leukemic cells, and you're actually looking at part of that bone marrow biopsy sample. Every one of the cells in the microscope field here is a leukemic myeloblast. She was treated but died 23 months after. She was normal cytogenetics, and her family gave us consent to sequence her genome, both tumor and normal. So with that and many other cells, or many other genomes sequenced, we sort of wanted to move this on. So I want to tell you about sort of how this fast forwards to, actually let me go back. So we learned a lot from this. We learned that you could do this, first of all, that you could actually sequence tumor and normal. We found mutations in this patient's genome, and it really started to sort of on the road to doing this for more and more cases. So we've come a long way, and I'll tell you a little bit more sort of where the research sort of stands. But we also wanted to be able to do this sort of in real time. And we had an interesting case that came to us of a woman who was diagnosed with AML at a hospital in the middle of Missouri, not in an academic medical center where they have all this, cool new stuff and so forth. She had her bone marrow biopsy, as all of these patients do, and they started her, the pathologist very quickly looked at the cells under the microscope and said, I see leukemia, should start treatment. They started her on chemo drugs. They also started her on that drug I mentioned earlier, Atra, because the pathologist said, the cells that I'm seeing, and you're actually looking at her bone marrow cells here, are very granular in appearance, and that suggests that she has this sort of M3 subtype that we can treat with Atra. So put her on Atra as well. Then the cytogenetics report came back and said she does not have a 1517 translocation. So there is no indication at all that she should be treated with Atra. And so Atra was discontinued, even though the pathologist thought that that was the right treatment. She was subsequently referred to the AML group here at WashU because she had persistent leukemia. And so they started her on treatment. But the physician, the oncologist who was taking care of her said, I bet she has M3 AML, even though she doesn't have the translocation. I bet there's something else going on there. And gee, can't you guys help us out here? Couldn't we sequence her genome and maybe find out what's going on? Because if she does have this fusion gene that's typically caused by the chromosomal translocation, we could give her the right drug and we could cure her. So we decided that we would do that. We sequenced her we sequenced her genome. Oh, just quick, here I'm just posing the question. Does she have APL? And if so, can we just give her Atra? Or is she going to have to go for a bone marrow transplant? So it was not only something that we'd want to know to best understand how to treat her quickly and cure her disease, but if she has to go this route, right, if you can't use this drug, if you have to go bone marrow transplant, a much higher risk case for her and her family, and much more expensive too. So we sequenced her genome, a little flap of skin from the area where they did the bone marrow biopsy, and then tumor DNA from the bone marrow biopsy. We turned this around in less than 6 weeks from the time we got her sample, which is unheard of. If you think about the Human Genome Project, the first human genome, it took us 10 years to sequence it, one human genome. So to basically get the sample, do all the sequencing and all the analysis in less than 6 weeks is nothing short of amazing. And what you're actually looking at here is from the sequencing, results. We've basically plotted her sequence across all 23 chromosomes here, chromosome 1, 2, 3, and on down to 21, 22, and X. And you can see some breaks in these lines. So these are regions of the chromosomes, chromosome 9 and chromosome 12, where this patient's actually lost part of one chromosome 9 and part of one chromosome 12 here. But the chromosomes that we were most interested in, based on what the oncologist's suspicions were, 15 and 17, we didn't see any indication of any loss of material, loss of DNA that might indicate that she did have this translocation. But we found something more interesting. So most of these patients who have this type of AML and have this 15-17 translocation, this is what happens. So in the normal cells, this represents the genes that are present on chromosome 15, and this represents the genes that are present on chromosome 17 in the normal situation. There's a break point, and you basically have a chromosome 15 break here and join to chromosome 17 here. The reciprocal thing happens as well, that part of chromosome 15 joins with part of chromosome 17. And what you have in the tumor, at least the result that's most important, is that the first half of this gene, called PML, on chromosome 15, gets fused to the back half of this gene on chromosome 17, which is called RAR alpha. Does that make sense? Good. And you even have some DNA that's deleted in putting those two genes together. The bottom line is that instead of having this gene intact and this gene intact, you now have this fusion gene, and the product from that basically causes that normal development of those particular types of blood cells to freeze early. So if you simply give them a drug, Atra, you overcome this, the cells mature, and everything is fine. Well, she didn't have that sort of situation. This was her chromosome 15, her normal chromosome 15, and her normal chromosome 17. These are the two genes that usually get fused by the translocation, right? There's a gene a little bit upstream of the PML gene called LOXL1. And what happened in this patient, if you can follow this, is that a 77,000 base pair interval on chromosome 15 essentially popped out of chromosome 15 and popped into a region on chromosome 17, right here where this little 5 base pair deletion is noted, just kind of like this. And so what she now has is, this is the deletion on 15. Here's the part of 15 that's jumped to chromosome 17. And amazingly, the first half of the PML gene has fused to the back half of RAR alpha gene, just like it does in the translocation. The problem with this, in this patient, there's no signature that the cytogeneticist can see, right? It's like the translocation is sort of like getting a small telescope, looking at the moon and saying there's a crater, right? To try to do this would be like taking a small telescope, looking at the moon and saying there's a rock. The resolution isn't high enough, you're not going to see it. And so the cytogeneticist can't see this kind of thing happening. So we were like, well, holy cow, this is pretty amazing. The end result is that she has the same problem as any other AML patient, and she can be treated with Atra. And so she was treated with Atra, she's in remission, disease-free, and it's a happy story. The correct clinical decision was made because we were able to sequence and determine this very unique oncogenotype and in a quick event. And we also learned that these sort of weird insertional events, and not just chromosomal translocations, can create these kind of fusion genes that are seen in many types of cancer. So we've gone on now and looked at other patients with the same disease that didn't show that translocation when the cytogeneticist looked at their samples. Guess what? More of the same. And so now we have new tests that we can use to basically ferret out these patients. So this is pretty fun, pretty exciting. And it obviously made a huge difference for that patient.